Thank you very much for joining the webinar today. I'm Nusha Chamar, head of Dell Laboratory Operations, and I'm honored to be a host of this webinar dedicated to very innovative technology in drug discovery. And these are actually DNA-encoded libraries, specifically a Dell platform at WuXi AppTec Kits. Allow me to briefly introduce WuXi AppTec Kits. This unit provides one-step target-to-hit service that enable companies in the pharmaceutical, biotech, and academics worldwide to seek premium drug discovery. As innovation-driven and customer-focused unit, WuXi AppTec Hits helps our partners improve productivity of advancing hit discovery through high-quality and cost-effective solutions. With industry-leading capabilities in protein science, DNA-encoded library technology, biophysics, and structure-based drug design, WuXi AppTec Hits is committed driving toward the realization of the vision that every drug can be made and every disease can be treated. To fuel the innovation in the early discovery in India, we are pleased to introduce WuXi AppTec's DNA-encoded library program to you. DNA-encoded libraries are seen as disruptive innovation in drug discovery, evidenced by multiple examples of Dell compounds entering clinical trials. I'm honored to introduce you the speaker, Alex Zatz. He has more than 14 years of experiences building and leading DNA-encoded library technology platforms and is currently the Senior Director of Dell Strategy and Operations at WuXi AppTec. Dell provides the means to discover novel chemical matter that modulates key biological targets, a common first step in drug discovery. Prior to joining WuXi AppTec, Alex built and led Roche Dell platform in Basel, Switzerland, and Nutley, USA, helped to develop the first industrial-scale Dell platform at Precis Pharmaceuticals, which was located near Boston, US, and later purchased by GlaxoSmithKline. Alex also worked as medicinal chemist at AstraZeneca, was NIH postdoctoral fellow at Harvard Medical School, and earned his PhD in organic chemistry from the University of California, Santa Barbara. In the field of Dell, Alex is also very keen to write different papers. He authored more than 14 peer-reviewed articles and different book chapters. So I'm really happy that we have an opportunity to listen to his webinar today. And please use the Q&A function at the bottom of the screen to ask questions. The questions will not be answered live during the webinar. The questions which will not be answered live during the webinar due to time constraints will be answered over an email. Do not hesitate to contact us at WuXi AppTec Hits at any time, or Hima Latha, who will follow up with you your interest in the technologies. So, Alex, the word is yours.
Okay, thank you very much. Yeah, so my name is Alex Zatz, and as Nuska said, I've been working in the Dell field for, actually, it's about 15 or 16 years now. So I continue to get older and to have worked in this field for a longer and longer period of time. And what I'd like to talk to you about, and I have enough slides for about 30 minutes, is about the Dell self-service kits that WuXi offers. So let me start off by talking a little bit about the difference between the self-service kits and our full-service Dell. So with the self-service kits, we send the Dells to you, and you can actually perform the screen in your own lab. What's nice about this is that it means that if you want to keep your target a secret, you don't need to share the name of the target with us. Another thing is, because you're doing the Dell screen in your own lab, it does save us at WuXi some costs, and we can pass along those cost savings to you. The alternative to the self-service screens, which is about what this presentation is about, is the full service Dell. In that case, you need to tell us what the target is, and you send the target to us, and we actually physically do the screen for you, and we help you understand what the data set means afterwards. But let's now just focus in on the self-service Dell. And let me first start off by trying to talk about the history of Dell a little bit, about how the Dells are made, why people do DEL screens, and about how the DEL selection itself is actually done, because I think if we cover those slides, the rest of the talk will make a bit more sense. So let me first start off by saying that today, almost every large pharma does DEL screens to find hits. That was not always true, though. I started working in the DEL field in about 2002. And if you look at this plot of years versus the numbers of papers which have come out on the DEL field, you can see that back in 2002, really up until about 2015, there was no one working in this field, and very few people were actually doing DEL screens. Since 2015, though, so this is in the last 5 or 6 years, the use of DEL has really taken off. Today, there's about 30 papers per year published in this field. And as I said, almost every big pharma and a lot of the smaller ones use DEL screens now in order to find hits for their drug targets. So why do these companies do DEL screens? The short answer for that can be found if you look through the lit a bit, and you can see a lot of published cases of DEL hits that have come out of DEL screens against targets, such as these shown here. We can also see what these hits look like after the hit is found and the med chem is done. So we can see what the hits look like straight from a DEL screen and what they look like after the med chem is done. And we can see the status of these hits. So for instance, a DEL that I made led to this DEL hit, which ended up in phase 2. Or this is a paper which we published at Roche of a DEL hit, which moved forward to become an in vivo probe. So this is why people run these DEL screens, because they have a track record of finding hits that can move forward. So what does a DEL library member actually look like? And here I'm going to show you this generic structure. It has a barcode where if you read off the bases of this code, you can tell what the structure was that was attached to it by way of what is actually a long, flexible linker between the structure and the code. I mean, here the linker looks like it's short and it's rigid, but it's actually not. It's a very long and flexible link. And so how are these DELs made? So they're made through split and pool CombiChem. And what that means, and I'll run through a quick example here, is that we can start off with a mix of 3 different things in a single tube. And every barcode encodes what the structure is. So if you take this mix of 3 different things in one tube, you can split them up into 3 different tubes. So we now have what we would call a split size of 3. And in each of these tubes, we have a mix of 3 different things where, again, you have a barcode, and the barcode encodes what the structure is. Now, a building block is added to each one of these tubes, and a tag is added to each one of these tubes where this tag encodes what this chemical structure is. And we now end up forming 9 new different structures, and where each structure is encoded by the code. You can then take these 9 new structures and you can again combine them into a single tube. So that's the pooling step. And so here we have shown a split and pool where we have a split size of 3, we create 9 new things, and then we repool them. So how do we make DELs rapidly using this? So the answer is, for instance, we could have 4 cycles of split and pool where each split has a split size of 96, and we can end up creating 85 million different new structures. Or we could have only 2 cycles of split and pool where each split size is 9,000, and again, we can end up creating 81 million new structures. Usually we at WuXi, we do prefer making DELs of this style where the number of split and pool cycles is small, because the physical structures of these hits or potential hits tend to fall more into drug-like space. But we certainly do make both types of DELs regardless. And at the moment, we've produced almost 200 DELs, and we can take all of our DELs and we can actually combine them into one tube, and we can screen them all at once. So how is this screen actually done? So we take the target of your choice that you want to find a hit for, and we immobilize it onto a solid phase resin. And then we take our DEL, which is a mixture of millions of different structures. Now, most of the potential ligands will not bind to this target because their structures don't match the binding pocket. And all those library members that don't bind, we wash away. But what we're left with are just the ligands, which can actually bind to the target, and we collect all of these. And we sequence the code, and from the sequence of the code, we can determine what the chemical structure was that bound to the target of your choice. And then we can resynthesize this without the barcode attached. And then we retest it to find out, was it indeed a real hit? And how potent was that hit? Right, so this is where we can start to talk about a bit. How do people in the drug discovery world or in the academic world access the ability to conduct their own DEL screen? So we have two different types of kits. We have a DEL open kit and we have a delight kit. So I'll start off by talking about what the DEL open kit is. This is for academic groups and it is completely free. And DEL open, although we at Wuxi, we produce the kits and we do all of the work, DEL open is actually owned and run by a rather large academic group. So it's a group of professors who oversee all of this. So it's not really an industrial thing. It's set up by academics for academic groups to be able to conduct DEL screens for free or at an extremely low price. And what's especially nice about the DEL open screen is that when and if you find hits in your academic lab, you can convert those hits into drugs, you could publish papers with them, and you can write patents with them. And you don't owe us or DEL open any money, milestones, and there's no strings attached. So how this works is that you order the kit from us, and the kit comes to you with 4 tubes in it, and you can run 4 different conditions all at once. So for instance, you can screen a target, and you can screen a counter target, and you can screen the target in 2 other ways. The screen itself is done in your lab, and then you send the samples back to us at Wuxi. We process this for you, and then we give you a report which you can look at and make a decision about, do you want to synthesize any hits from this screen? You do need to pay for the synthesis of these hits, and you're not provided the structures of your potential hits, just how they enrich. We then send the hits to you. You test them in your own lab, and if the hits look good, if they're active, then we will give you the structures of these hits at no charge. And you are free to do whatever you would like to with these hits. But remember, DEL Open is run by an academic group, and they're about sharing data. So in exchange for us sharing you the structures with you, we require that you share the name of your target and the metadata from your screen, which then goes onto the DEL Open website, so that other academic groups can actually see this data and possibly work with you to produce more hits. And so this is an exchange where we exchange the structures of your hits in exchange for the data so that other academics can look at it. Now, in addition to the DEL Open Kit, we have a Delight Kit. And the Delight Kit is more about startups and companies wanting to find drugs. And how the DEL Kit works is you pay a fee up front, and then you get our kit. Again, you run the screen in your own lab, and you send the samples from the screen back to us, and we process them, and then we share the results of the screen with you, but we don't show you the structures of the hits. You just need to make a decision. Did the screen work? And if you think it did, then you pay an additional fee and we unblind all of the structures for you. And so we might send you a list of 100,000 structures, all of which were enriched in your DEL screen. You are then free to do anything you want to with those hits. So you can follow them up, you can publish patents, you can discover drugs. There are no other milestones. All right, so we have a few numbers here to share. One is that thus far we have sent out 346 kits globally. So a lot of people have ordered these kits thus far. Because there's a lot of different structures in each one of these kits, we have effectively run over 300 quadrillion assays from these kits. And also, we have unlocked 17 million different structures. So we have sent out 346 kits, and a lot of those times our Delight partners have decided to pay the fee to unlock all the structures, and we have shared over 17 million structures with them. So this is good news. You might be concerned that if we have shared so many structures that there might be nothing good left in these kits. So let me try to address that with a couple more slides. First of all, the Delight kit has over, I believe it's 15 billion different structures in it. So far we have only shared 0.0008% of all the structures in our Delight kit. So although we have shared a lot of structures with a lot of partners, the vast bulk of the structures in our Delight kits have not been shared with anyone. So there's a lot of stuff left in there. So we have just released the newest version of our Delight kit. This came out about 2 months past. We update the Delight kits every year. And right now, the new Delight kit has 58 DELs in it, and the new DEL Open kit has 30 DELs in it. So as I mentioned prior, we can produce 58 DELs, and then we combine all those DELs into a single tube, and you can screen that one tube all at once. And that means that we now have over 15 billion structures in the Delight kit and almost 5 billion structures in the DEL Open kit. So whether you use a DEL Open kit and it's completely free, or you use a Delight kit, you do have to put in the time to conduct the screen. The screen itself will take you about 1 day to physically do in your own lab, but it does take some time to get ready to do this screen. And we, on our side at WuXi, we have an obligation to make sure that when you send the samples back to us, that we process them properly. So towards that end, we have created an automatic workstation to help us do this job better. And what I'm showing you here is data where we have processed the same 16 samples multiple times by multiple people over multiple days and then sequenced them. And what we find when the sequencing is done is that no matter who processes these samples or on what day, the results are always basically exactly the same. So you can be assured that when you send the sample to us, that no matter what day the sample comes in or who it is who processes the samples, the results of the screen are going to be the same every time. There's also a nice thing that we've added on our website. So you are now able to go onto the website, and after you've sent the samples to us, you can track the status of the sample. So you could tell that, in the past, you might have been a little nervous because you didn't know if the screen worked, and then you put it into shipping and you don't know if we've gotten it and you don't know what step it is. Now you can go online and you can automatically find these things out. So you can go onto the website, you can register, you have a password, and you can sign in anytime that you would like. And you can determine, have we received the shipment? You can determine, has the qPCR been done or has the sequencing been done? And then really the nicest part here is online, you can look at the results of your screen, and I'll talk about this in a later slide, and you can actually interact with your own data. So let's talk a little bit about the interaction with your own data and why that is important. So in the case of a Delight screen, you send the samples back to us. We process it. We generate a report for you. But then you need to look at this data and you can't see the chemical structures yet. And you need to decide, do you want to pay this fee or not in order to unblind all of those structures? And you might be very picky about what you consider to have been a successful screen. And so how are you going to make this choice about, do you want to pay this fee and do you want to unblind this data or not? And so the answer is you can go online and you can see data such as shown here. So for instance, let's say in condition 2 is your target of choice, which you want to find hits against. So we're calling this region A. And you've put in a counter target in condition 3 and in condition 4. So you want to find hits that show up in condition 2, but do not show up in condition 3 and condition 4 or any overlap between them. So you can type in here and you can say, well, only show me potential hits that have enriched in condition 2, but don't enrich with an enrichment of 10 or less in condition 3 or condition 4. And then it gives you the result of this. It says, well, you have 5,200 possible hits across 47 DELs. And it gives you a line plot for each one of these, where it's showing you the enrichment level, and it's showing you the enrichment in each of the 4 different conditions. So you say, well, 5,200 hits is a lot. I want to be a little bit more stringent. So let me ask the question, can I find hits that enrich at a level of 100-fold in condition 2, but then less than 1 in condition 3 and 4? So basically they enrich strongly in condition 2, but have absolutely no enrichment against the counter targets. And the answer then is we find that there's 2,000 potential hits across 20 DELs. And again, we see each one of these hits here in a plot. For instance, we see a hit which has 0 enrichment in condition 1, enriches in condition 2, and has 0 enrichment in condition 3 and 4, the counter screens. You could also look at this information even more, and you could say, well, I only want to see hits that have a molecular mass of less than 500 grams per mole, and I only want to see hits that have a C log P of less than 5, and you can then see how many hits you have left. And from that data, then you can make a choice. Do you want to pay the fee where you actually see all of the structures so that you can work on those structures and start up your drug discovery program? So we have 3 different sites around the world where you can send your samples to for us to do the work on them. So you can send them to Shanghai, you can send them to Boston, or you can send them to our Munich site. Depending upon where the physical screen is actually done, you can choose which place makes the most sense to send it to. If you have a collaborator who is conducting this screen for you in the US or within the EU, then it might make sense to send it to the Munich site or the Boston site. Also, at our Boston site, you can actually physically go to our Boston site and run your screen in our lab. So if you have an academic partner in the US, that might be an option. Otherwise, you can simply send the samples to us in Shanghai and we can process them there. So there's one more part of this, which is when you run a DEL screen in your own lab, at least in the past, before you sent the samples to us, you never knew, did the selection go well? Running a DEL screen isn't that hard. We provide videos for it, protocols and other things to help you, but we have found that often academic labs tend to have a harder time doing the DEL screens properly, whereas the professional labs, the startups, and the biotechs almost always do a very good job. But what we include now in our new versions of these DEL kits is the ability for you to do real-time QC while you're doing this DEL screen. So for instance, and this is especially true in some of the academic labs, we send you 4 different tubes, but what we find is that there's a lot of mixing up of these tubes. So some of the sample from tube 1 will end up in tube 2. And so we have sent you a QC test that you can do before you send the samples back to us to make sure that samples aren't being mixed up. The other thing which we now allow is for you to test the amount of DEL which you recover after each round of the selection, and to make sure that the amount of DEL is the correct amount. And again, we find that in the professional labs, this has not been an issue, but in some of the academic labs, this has been a problem. And so if your selection doesn't appear to pass these QC tests, you could talk to us and you can make the decision of, instead of sending the samples back to us, you could go online, order a second kit, and run the selection again. This time, try to be a bit more careful and make sure that this cross-contamination issue doesn't happen. Okay, so this is my last science slide here. So to sum this up, on these new DEL kits, what this allows you to do is to run the screen in your own lab and to do the real-time QC while you do this screen, so that when you ship it to us, you can feel very confident that the screen went well. You can then go online and you can track the shipment to us, and you can track the process of us working on your kit in order to get you the data set. And then when the data set is ready, you can actually go online and you can interact with your data set to make the best judgment that you need to make to decide, in the case of DEL Open, which hits do you want to resynthesize? And in the case of Delight, do you want to pay the fee to unblind all of the data? Now, we also have a partner with Schrodinger in the US, and you do have the option of interacting with them and having us send all of your data to them, where they can then use ML in order to process your data for you and help you choose which hits you would like to follow up. But they can also data mine external vendors in order to find potential hits that you could purchase from those external vendors and then test on your target of choice. So if you have an interest in that, that option is always open too. Okay, and with that, that's my last slide. And we can now enter into the Q&A. And thank you very much.
Thank you, Alex. And thank you, the audience, for contributing already quite some questions. I still encourage to, let's say, contribute more. So I will try to group the questions according to the topic. So let us go first to 2 questions, which actually are targeting practically how the DEL functions. One question is, if there are millions of molecules, how accurate will we find target binding to molecule?
So historically, what occurs is that we'll run a DEL screen. We'll choose 20 hits that we think look the best. We will resynthesize those 20 hits, and then we'll test those 20 hits in a follow-up assay. Very often, the follow-up assay will be a biochemical screen. And hits that show up active in that screen at, let's say, a micromolar or better will show up as active hits. Of those 20 hits that you resynthesized, you can expect about half of those will be active in the follow-up screen. Now, that doesn't mean that the other half don't bind the target. There's usually a window of how potent a hit needs to be in order to show up in the follow-up assay. But if your follow-up assay is different from the DEL screen itself, about half the hits will end up being active. So that's just the historical data. Some targets, the hit rate is much higher, and other targets are really tough targets, and the hit rate can be much lower.
So combined with this question, it comes the next question. If we want to work with structural analogs, how does that work in this case?
Structural analogs. So the Dell screen itself has a tendency to show some chemical SAR. So for instance, if you run a Delight screen, you might find a cluster of hits, all of which have similar structures. Now, in the case of Delight, we send you a long list of all of the potential hits, and this list might be 100,000 structures long. And how you choose to follow those up, how you choose to do the MedChem SAR is really up to you and the target team. You as the target team, you might have other hits from an HTS screen, which sort of help guide you. You might have, you might know about other hits from other patents or from other sources, and that might guide what SAR you follow up, or you might know absolutely nothing about the hits that might hit that target. But that's really up to the target team. Whereas, you know, that's not up to us, people who run the Dell screen.
Thank you, Alex. So here's now practically now there are a set of questions regarding the costs and timelines. Mm-hmm. So what is usually typical turnaround time for NGS sequencing and report submission? Yeah,
so when you send the sample to us, we will give you the report in about 4 weeks. So it's quite fast.
Couple of questions are related to the cost. So what is the approximate cost of the light kit? Mm-hmm. And also there's a question if there is any royalty fee subsequent disclosing the structures.
So first, the royalty fee is easy. The royalty fee is there is no royalty fee of any kind, and there's no strings attached to any hit. So whether it's Dell Open or Delight, once you see those structures, you are free to work on them, discover a drug, you can make billions of dollars, and there are no fees to be paid later. As far as the cost of the kits, we can always provide that to you by way of email, but I don't think I'm allowed to share costs during a webinar. So the exact cost I couldn't give right now. But if you send us an email, and I think this is the email down here, I can send you, or someone will send you the cost breakdown for the Delight kits. It's a clear-cut cost, so everyone gets the same exact cost.
Yes, and if I may add, also when you log on the HIT-S webpage and you check, for example, under the Light kit, you can practically order it on the webpage, and there immediately the price will appear.
Yeah. So I think the price is actually online, but still I'm a little hesitant to toss out.
Yeah. So, but there are resources. So, and in the combination with this question, there's additional one. How is confidentiality maintained?
So we don't know what the target is at all. So you don't share the target name with us. So there is no ability for us to break the confidentiality on the target. The data itself is confidential and will never be shared. In the case of Dell Open, of course, the same rules apply. And in exchange for the structures, that is what you then hand over to us. We are then allowed to publish the structure and the metadata of the selection on the Dell Open website.
Next two questions relate directly practically to the wet lab. So how many selections one can handle with this kit? For example, target, anti-target, and both in presence and absence of potential binder. And as a continuation of this question, what is the infrastructure required in the lab to do the screen?
Yeah, so the kits have 4 tubes. So you have a total of 4 conditions. One of those conditions will need to be the magnetic beads by themselves with no target added. So that leaves you with 3 conditions that you can screen. So you could do your target and 2 counter targets, or you could screen 3 different targets with 1 kit, or you could screen a target with a known control that blocks a binding pocket, and maybe a different construct of that same target. So it's up to you how you want to do that. We can help you a bit in helping you decide how to design that selection. But again, you don't need to share, and actually, we ask that you don't share the target names with us in the case of the Delight kits or the Dell Open kits. Oh, and what equipment do you need? Very little. I believe you can run the Dell screens with practically just pipetters. Nuska, you might be in a better position to add to that. Do they need an automatic pipetter? Is that a requirement?
No. So this is really the performing affinity selection in the lab does not require any specific equipment. So you need, let's say, a typical equipment for molecular biology lab, a centrifuge, a heating block, manual pipetters, maybe magnetic, a magnet for magnetic bead separation. If not, you can use also centrifuging. So this is really very basic. So to learn more about what is needed, I invite you to practically to visit our webpage, where you can actually already register on the Dell on air. And with this registration, which is for free, you get access to our resources, where you can also check the videos, how selection can be performed, where you can also read our protocols. So there you can really find a lot of resources, which can help you to understand better what you need in your lab. Okay, so next question is actually, does it work with membrane proteins, and what is the success rate?
So it can work with the membrane protein if you can get it into a soluble format. And in that case, the success rate can be quite high depending upon what the protein is. But will it work with membrane extracts? The short answer is no. And will it work with like cellular screens or things along those lines? Again, I think your chances of getting that to work properly with the Delight kit is extremely low, unless you have a lot of experience with it. So if you can get a detergent solubilized and stable membrane protein at a relatively high concentration, the Dell screen is run in exactly the same way as for any other target, and your hit rate will be the same as it would be for any other target.
What is percent hit rate for any protein target? How is
that?
Just
on, I mean, it ranges from 0 to 100, but it's, I mean, for a good screen that worked well against a target that is stable, I would expect half the hits to reconfirm in the follow-up assay. But again, bear in mind, the reason why the confirmation rate is 50% and not 100% is because you're usually following up the screen with a biochemical assay. And not all ligands that bind to your target will be biochemically active, and not all ligands will be active enough to show up in that assay. So that's the reason why the hit rate for a good screen that works well against a good target tends to be about 50%.
One of our attendees is curious how the buffer depends about the buffer influence on the hit rate. What is the percentage of false positives?
Yeah, so that goes back. I mean, 50% of the hits will be active in your biochemical assay follow-up. But again, something not being active in the biochemical assay doesn't mean it's a false positive, it just means that it's not biochemically active. Bear in mind, DEL is a binding assay, right? So you could find hits that bind outside of the biochemically active pocket and don't have a biochemical effect, or at least not one that you can pick up in the particular screen that you run. Now, what is the importance of the buffer? Well, it's very important, like any binding assay. If you choose the wrong buffer, the protein won't be happy. And if the protein isn't happy, the screen won't work well. So we generally advise that you find a buffer that works well for your target. And that buffer might be the same buffer that you do the biochemical assay in, or that you run the SPR in. So we would generally suggest that you use that buffer because you know the protein is happy underneath those that circumstance with those buffers. I'm going to go off a little bit on a tangent and just say, if your protein is misfolded, if your protein isn't in its active state, then the result of your screen won't be any good. So DEL usually requires crystal grade, pure, properly folded target.
And also one of the attendees wants to know if there are any restrictions on the use of detergents in the buffers.
On detergents.
Detergents, yes.
None that I'm really familiar with, no. As long as the protein can be immobilized onto the solid phase resin, so you could use a streptavidin bead, you could use an IMAC bead, as long as the protein capture works and the detergent doesn't prevent that, then any detergent could feasibly be used. It would be perfectly fine.
After the data analysis, are the structures that are disclosed to other companies subtracted from your library, or they continue to be there?
No. So in the case of Delight, we don't know what your target is, and we have no idea which of the hits you follow up. So there is no ability to remove structures from Delight. And so structures that you find, someone else could have run the same target the prior month and have found those same structures. We don't keep track of this, and we don't remove structures that have been shown. Yeah, so that's the short answer.
Thank you. So this is one very interesting question. Another interesting question. This attendee would like to know how our libraries compare with other players in this field.
Yeah, even if I knew the answer, I mean, I don't talk about other players. So now I know them all. I will drink with them and such, but I don't talk about their stuff.
Okay, so this is actually all the questions from today's session.
Oh, there were a lot of good questions there, I thought.
Actually, one came in now.
Oh.
Can we send samples for Dell Open kits after screening to Indian labs?
Oh, say that again. Can we
send post-selection samples? The question is related to Dell Open.
Okay.
After screening to Indian lab for NGS processing.
No, so we don't currently have a processing capability within India itself. So my guess is the easiest place to ship those samples would be Shanghai, at least in terms of physical closeness. But I must admit, I don't know about what paperwork is involved. Paperwork wise, maybe it's easier to send it to Munich or to Boston. That I don't know. But the samples after they're done at the moment would have to be shipped to the Munich lab, the Boston lab, or the Shanghai lab. If there were a lot of people using Delight kits and Dell Open kits in India. I mean, we could think in the distant future about having a site there that this work could be done in, but it's not going to happen anytime soon. So I wouldn't count on it. And I think one more person just raised their hand. Something popped
Yes,
up.
exactly. I got the Q&A here, the question. What is the processing time per sample? And of course, they are concerned that the cost will increase when shipping has to be done on ice. So there are two questions.
Yeah. So there are some, the cost of shipping on ice is not good. It could be $1,000 or $2,000 to ship it, I think, on dry ice. Although I suppose it depends where you're shipping it to. The processing time is 1 month. So you run the screen, you ship the samples to us, and we need 4 weeks to do the QC, the PCR, the sequencing, and generate the report, and then to share it with you. That's a 4-week time.
So thank you very much to all to contribute these very interesting and very good questions. Thank you, Alex, for a very detailed presentation of our Dell Open platforms and the Light platforms. And I invite you all to reach out with any additional questions, concerns, details which you would like to know per email, and we are more than happy to address these questions. Thank you very much again for your attention, and have a great day. Bye.
Bye.